For our next session, we have Dr. Chanli Valiathan. Chanli is the Director of Modeling and Decision Sciences at Janssen Pharmaceuticals, where she leads the model-based meta-analysis group. She built this group from the ground up, planning and executing on the vision to expand the use of historical clinical trial data for key strategical clinical decisions across the entire Janssen R&D portfolio. Dr. Chanli is a seasoned veteran who has worked with GSK and Merck during her vibrant career. Today, her talk will explore the various stages of compiling useful datasets from published data all the way to leveraging this clinical knowledge to generate insight and enable data-informed decisions. Dr. Chanli?
Thanks. Can everybody hear me okay?
Yes.
Okay, great, thanks. Thanks for that very nice introduction. I'm just pulling up my slides here. As Miriam mentioned today, it's really great to be here and to be able to speak to all of you about some of the work that we're doing, at Janssen in leveraging data to actually inform clinical trial design questions. And so today, what I'll be going into is some of the challenges and opportunities that we see in unlocking insights from published clinical trial data. So let's get right to it. So before I go into the entire data component of the talk, I want to describe what we use the data for. So as Miriam mentioned, I lead the model-based meta-analysis group. So today I'll give a brief description of what that means. And then we'll sort of explore some of the different types of clinical data that are available for modeling purposes and just, do a quick check. Are they all accessible and what are the challenges around that? And then dive into some of the, again, the roadblocks and roadblocks can mean opportunities for all of us to create solutions in creating fair data sets from published literature. So what is model-based meta-analysis, or MBMA? MBMA is actually a tool that allows us to integrate information that's available in published clinical trials to really sort of pull together everything that's known about different compounds tested in specific diseases so that we can build knowledge. And through the modeling work, we can actually compare all of the effects of different treatments across different trials, across time, and really understand the entire landscape of a disease in terms of treatments and clinical endpoints. So this figure here shows sort of the process of how we go about doing this. We start with unstructured data, and that unstructured data is, usually it's PubMed, sometimes clinicaltrials.gov. So basically, it's just text, and we usually have to work very closely with the clinical team to identify which subset of those papers we're actually interested in, so that we can then build the models on top of that specific data set. So coming back to what Abhishek was talking about, we need the right data, right? And before we need the right data, we need the right questions. So we really need to understand what the clinical team needs. And so we define the disease area of interest, the treatments of interest, the populations of interest, the clinical endpoints, efficacy or safety endpoints that are of interest, and then various biomarkers and so on and so forth. So once we define all of that, we're then ready to identify the publications from PubMed that have all of those characteristics. And then basically there's a huge process in converting all of that unstructured data into structured data so that we have a custom database. Now, once we have that custom database, the model development can start. And the advantage of using models is that, not all clinical trials are designed the same way. The populations might be different. the design of the trial might be different. And so through modeling, we can actually account for those differences and start normalizing across those various factors so that we can do more apples to apples comparisons. Then all of that information can be used to inform clinical design and development questions. So these MBMA models, I do want to highlight, they're not specifically AI based at the moment. Everything we do are more mechanistic or empirical models, but you can imagine or envision, as we are able to compile more and more data sets and different types of data, that these MBMA models could also be AI-based models. So why do we do this, right? What kind of questions can this type of modeling help answer? I've tried to highlight a few different ways that we use these models. So the first box there, it shows a network of how we can represent the data that we have. So, each circle there, each node is a particular treatment that's available in our data set. And the size of the circle represents how many patients or subjects have been treated with that particular compound. A line between the two nodes, connecting the two nodes, means that there was a clinical trial that actually tested those two compounds head to head. And the thickness of the line shows how many times that sort of trial was conducted. So basically, you can think about the entire data set that we pull from literature, we can represent in this network. Now, the advantage of doing that is that once you have a new compound that you've run a phase 2 study, you have new data, you can actually plug that into the network and then do what I call node hopping, so that you can start comparing your new compound to any other compound that's in the network, as long as they're connected through nodes. And this is actually very powerful, because by doing this, you can have head-to-head comparisons of your new compound against something that you have not really tested in the clinic. So you're basically doing a head-to-head in silico comparison, but that gives you a lot of information on how well your new compound will perform and your probability of success, for example. Now, you don't really need a compound to do this analysis. You can actually just, before you have any compound, you can just chart the competitive landscape in a particular disease area. So if there are many treatments already approved, is there any gap that you can sort of jump into and cover? And those type of questions can come up, especially when we're exploring collaborations or performing due diligence work. And then the third box here is around clinical design. So a lot of the work that we do falls under this bucket here, where we use historical trial data to help benchmark what we will see in the next trial. So for example, if you have a comparator or a standard of care that you're using as your comparator arm, you want to know what that effect will look like in terms of the clinical endpoint of interest, and the distribution around that based on historical data, how variable that value is. And then basically beyond that, how much confidence do you have that your new treatment will be better, and what is, and by how much, and what is the variability that you expect around that. So all of those kind of numbers that we can get from historical data help with sample size calculations, probability of success calculations, go, no go decisions, and so on. So these are just a few examples that I've given here. There are many other examples of use cases where we've used this historical data to really drive decision making for our programs. So, I think all of us here sort of know and understand this diagram to a certain extent. We always start with raw data, and mostly, we aim to end up with this enabling informed decision making. But through developing the models, we actually build out incorporate the knowledge that we know, and you saw examples of that with Abhishek's slides as well, where the models actually capture, for example, the mechanism of action of a treatment, and also study-to-study variability. And so everything is accounted for with your model. And then once you have that, you can actually perform simulations and really show the clinical team what all of that knowledge means, right? So you can actually simulate out new scenarios and really give a 3D view to the team members of what all of this data means. And it's really helpful for scenario exploration, especially when you're trying to go into a new population that hasn't been tested before, and so on. And so in that way, we can drive data-informed decisions. Again, the likelihood to be a market leader, for example, which is without all of this data, very difficult to quantify and have a concrete grasp on. So now, we'll switch gears a little bit and start talking about the data that goes into all of this modeling. It's all well and good. There's a lot that can be done with the modeling, but we have to start with the data. So, there are different types of clinical data that are available. Some sources are more accessible than others, right? So, as a pharma company, we do clinical trials, and we have a lot of internal clinical trial data from our previous trials. And all of this is usually in a very nice format, the standardized format. You can have scripts that pull out information that you need. Everything's tracked, and for good clinical practice purposes, all of the personal identifiable information is protected, and we follow all of the legal and regulatory requirements in handling this data. The challenge there is that is proprietary data. So of course, one company will not have access to the same type of data from another company, right? It's all proprietary. So in certain sense, it is accessible to folks within the company, but not outside. There are other types of clinical data as well. So there's insurance claim data. So now this is in the real world space. So we're not looking at a controlled trial or anything like that. It's just what happens in the real world with comorbidities and in an uncontrolled fashion. But oftentimes you can purchase these data sets. They are standardized. You can query them. It's in a database format, and also the personal identifiable information is protected. And one thing that I must note is that many of these are individual level data. And that is important because when we come to the published clinical trial data that we mostly use, it's all at the aggregate level. So many of these issues about identifiable information, we don't need to worry about. But in a general sense, even the insurance claim data is, pretty, I want to say tractable, right? And then you have sort of biobank repositories, TCGA, repositories from the Broad Institute, and all of that again is easily scriptable and accessible. Some you have to buy, others are free, but all of these I feel are again very nicely structured and tractable. However, when you start looking at published clinical trial data, it's the wild west to a certain extent. It's all unstructured text or text format. The data is not individual level, it's aggregate level. Depending on your subscription rights, the publications are free or paid. But basically, this sort of data set is very, intractable, not easily accessible. And this is sort of the area that we are working with. So what does it take to make all this published data fair, right? Findable, accessible, interoperable, and reusable. So, it actually takes a lot of time and effort to transfer all of that knowledge that's trapped or locked in the publications into a structured database. And we have to go through various stages of, identifying relevant publications, and within those publications, what specific numbers do we actually pull out? Most of the work is done manually, and this is where I think something like Polybird that Abhishek mentioned might be very useful in this sort of scenario. So definitely opportunities, again, I'll come to those in later slides, but there's a lot that goes into creating these structured data sets. And even then, you'll see, it's not a final data set. We still need to work on these structured data sets to make them model ready. But the main point is that it's not easy and it takes a lot of time and money. So the first step really is, we have to identify the relevant publications. And so this is something that we have to do very closely with the clinical team, because again, you need to identify the right question, and that drives the right data. So, we really have to understand what the clinical team needs, what efficacy endpoints are they interested in, what population, what treatments, which trial phase, for example. And, it's this step, it might seem simple, but it's actually very nuanced, because when you're creating such a database, you're putting in so much time and effort. You want to make sure that the database is general enough that you can actually ask multiple questions of it, because oftentimes as you start working on a problem, the questions also kind of change. And so you have to be cognizant of those changes that may occur and create this search term in a little bit of a generalized way. But at the same time, you can't make it too general, or you'll end up, spending a lot of time and effort. So although this first step seems very simple, it actually does take a lot of thought to finalize that search term. Now, once you do finalize the search term, you ping PubMed, and what you'll get is a list of thousands of abstracts, 6,000, 10,000, and so on. So how do you find, not all of them are relevant, right? So how do you find the ones that are relevant? So usually, you have a team sort of reading through abstracts and trying to figure out, yes, this is relevant or not. And then you always have to kind of capture the reason why. And it's done, again, not just one person, but multiple reviewers at the same time. And then if there's consensus, you're all golden. If not, then you have to find a subject matter expert to sort of break the tie, or like decide which way to go. And this is actually quite challenging to do. It's again, time and cost intensive. And, it's not as easy as just doing certain keyword searches in the abstract. Sometimes, for example, in oncology, you have all of these nuanced ways that the abstract is written, where you might want, a population that's in the second line of therapy, but then, you might see that the abstract says second or third line. So do you include that or not? It's not just about finding the right search terms. And this is where I think there's an opportunity to improve, and where NLP and not just bag of words or entity recognition, but more understanding the context of the words can help and really speed up this process of identifying the relevant publications. So again, this is, I think, highlighting, again, opportunity where some of the advances that we've seen in the past few years can help us identify the right list of publications. So once you go through all of this, you end up with, say, hundreds of abstracts instead of thousands. And once you have those, it's still not, that trivial, because all of these are actual publications. So the data is actually contained in figures, tables, and text. So again, usually you have people, manually reading and extracting the values that you want. Remember, the database is not curated on all of the numbers there. You kind of are also picking and choosing which endpoints you're interested in. And again, this is another place that there may be an opportunity to improve, because, we have methods of digitizing data. So can we automate that, extracting information from tables and text? We saw an example again earlier with Abhishek, where the NLP algorithm was pulling out, specific values and information from text. But how do we do that in a consistent and automated way? Of course, we'd need to put an effort to actually create all of that for our database of interest. It's again, I think there are many opportunities that I'm highlighting here that are really, ready for innovation that can speed up these processes. So once you do all of this, you end up with a structured database. And now that is really where the modelers come in. But even then, right, even with a structured database, we're still not model ready. There's there are always challenges that arise, and these are more nuanced. It's difficult to define something that works for all data sets, but in general, there might be issues of data quality, incorrect or missing entries, duplicate records. Sometimes you want to understand what data is relevant. Again, this goes to talking with the clinical team, and then there might be some studies that have like a different design that wasn't very obvious when you pulled or created the database. So now you have to go back and read those papers and really sort of parse things out. And some studies might report the data early on, and then you have updated data that comes later on that might end up as duplicated records in the data set. And once you have data that's missing, what do? Do you just throw out those studies, or do you do imputation? And so there are all of these different types of questions that still have to be answered on top of everything that you've created. So again, coming back to, I think we all know and understand this, right? So in that whole process of starting from raw data and ending up with data-informed decisions, while the weight is often on the decisions and the impact comes presumably from the data-informed decisions, actually a lot of the time, effort, and cost needs to be put in upfront in actually getting the data right. And then, the models can happen, can be built on top of the data, the simulations, and so on. But this data component is actually just very important to get right. And I think I'm preaching to the choir here when I say that. So this is basically my last slide. So hopefully I've shown you, the value of using published clinical data for decision making. And we've talked about the challenges that still exist. I do think that we are positioned well to start using some of the NLP and image processing techniques to help with these curation questions. But at the moment, it's still the biggest sort of time cost investment that's needed is really in that data set creation for that whole process of a data-informed decision. So at the end, it's still worthwhile, but there's always room for improvement through automation and so on. With that, I'll stop, and I'll thank the organizers again for letting me speak here, and I'll take some questions if there's time.
Okay, thank you very much. I very much enjoyed your talk, especially when you mentioned the need for having the fair data and all the efforts needed, all the time needed to actually spend time and get in there. I very much enjoyed it. So I'll go through one of the questions that is from the audience. The question is, what are the benefits of using MBMA over other meta-analysis techniques?
Yeah, so I think that's a great question. So general meta-analysis uses statistical methods. So for example, if you have data for a specific clinical endpoint, say it's an efficacy endpoint at 16 weeks, for example, then with meta-analysis, you can pool information that's reported in all the publications for that specific efficacy endpoint at 16 weeks and summarize it through meta-analysis. However, the papers that don't mention any endpoint at 16 weeks get thrown out. And you're also not able to account for covariates. So if there's a paper that looks at, the average age was 65 versus another paper trial was 55, you kind of pool all of that, or you decide to throw one or the other out. With the model sitting on top of all of the data, you can actually, through the modeling, even the paper that didn't have the 16-week data gets included because if you build out a longitudinal model, you can actually account for the time points that were earlier on and sort of leverage all of the data to then predict what would happen for that child at 16 weeks. And then also with the age differences, if you have age as a covariate in the model, then you're not throwing out the data, you're using all of it, and then projecting out what would have been seen in that child for an age of 65 years old. So this is the advantage of using model-based meta-analysis rather than just meta-analysis.
Awesome. Thank you very much. Thanks for your talk. We really enjoyed it. I'm sure everyone learned from it.
Okay. Yeah, thanks all.
Of course. Thanks.